everyone, and welcome to today's webinar, Multiplexed Lateral Flow Assay Development, Benefits, Risks, and Strategies for Success in Infectious Diseases and Oncology. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Fortis Life Sciences. To learn more about our sponsor, please visit fortislife.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would now like to welcome our speaker, Andre Alfaro, Senior Director of Assay Development, Fortis Life Sciences. Andre, welcome. You may now begin your presentation.
Hi, I'm Andre Alfaro, and today I'm going to be talking about multiplex lateral flow assay development and really highlight the benefits, the risks, and give you guys some strategies for developing these assays as it pertains to oncology and infectious disease, and all assays in general. Now, this is just a 20-minute presentation, and I'm not going to be able to provide some recipe for success, but what I can do is hopefully provide some guidance on what was done in the past, experiences that I have, to really help you navigate this journey. If you have any questions post-presentation, feel free to reach out to us at nanoComposix and Fortis Life Sciences for any questions you may have. So as a reference for the conversation today on multiplexing, I wanted to use infectious disease and oncology as that point of reference. So here I wanted to illustrate where those sectors are today and where they're growing to be tomorrow. So infectious disease, for example, and we're all very aware of it with COVID-19, that industry as of now is about 28 billion. We expect that to grow to about 51 billion in 2030. In the oncology sector and cancer diagnostics, we're about 17 billion in 2021 and expect that number to be almost twice that in 2026. As an example that I'll be referencing throughout today's presentation, I want to bring up a project we're working on with Vigilant Biosciences. Vigilant received breakthrough designation just this year from the FDA for a fully quantitative multiplex saliva oral cancer screening test. This is the highest complexity you can imagine for a multiplex assay. Difficult matrix, multiple targets, fully quantitative. And because of the complexity of this, I'm fairly confident that depending on your application and intended use, I think you'll find that you can be able to be successful in this field, particularly with the FDA becoming more open to the idea of salivary diagnostics in the point of care and OTC spaces. Let's define multiplexing. So by definition, a test intended to measure one or more analytes in a single device is multiplexing. Now, there's a lot of ways that you can actually accomplish this. So here on the screen right now, you're seeing 3 examples of that. And there's also a multitude of others that are not listed here. So on the left, you can see a single strip with multiple targets per strip. In the middle, you can see multiple strips for a single target per strip in a single device. And on the right, you can see multiple strips with multiple targets on each of those strips. Each of these are constituted to be multiplexing. And really the avenue you go for, depending if you want to go on with the example on the left or the right in the extremes, are really based on your target product profile. The complexities and obstacles you face will determine the route you want to go. Now, every assay is not going to fit on the ideal model, which is on the left. Because of cross-reactivity, technical obstacles with dilutions, there's going to be some considerations there that will force your assay to either be split or removed. So these are different examples of how you can overcome that without having to lose that really important biomarker that you've selected. So why multiplexing? What are the benefits of this approach? Now, everyone has different input on this point. I really try to break this down into kind of 4 talking points: clinical and analytical performance, value proposition to you, the client or the developer, the intellectual property that you can create from it, and manufacturability. So let's talk about the benefits that I just listed, starting with clinical and analytical performance. I've listed some of those benefits, what they can be: clinical sensitivity and specificity, analytical range and semi-quantitation, sample volume controls. So what does that mean? So clinical sensitivity and specificity are usually derived from what you're detecting, what you're not detecting in that assay. So to improve sensitivity, for example, for COVID assays, when you needed to detect low TCID50 samples or low CT counts, adding in antibodies to, say, the spike protein as well as the nucleocapsid protein helped with that because it increased my available binding. For certain applications like breast cancer reoccurrence, in order to detect all the cohorts of the disease population, you might need more than one biomarker. HER2 for HER2 positive samples will get you pretty far, but perhaps a second marker like CA15 or EGF will help you with those certain cohorts in order to increase your sensitivity and specificity. For analytical range and sample volume controls, we've developed products that were in such high doses and didn't allow for dilutions. How did we overcome that? We actually use multiple test lines for the same target to bin results. So if a line developed on the first position, it would be above X, on the second position above Y, indicating almost like a semi-quantitation detection of the assay and target analyte. This is more used for drugs of abuse and sample controls. So a lot of the LAMP assays we've developed had internal controls to ensure that the reaction took place upstream. That's really important to do, and to do so we use the multiplex assay format to do that. Another benefit is value to the customer. Now, as a consumer, just like you or I, what we're looking for is all the information we want out of this test. Am I sick? Am I not sick? At a cost point that's effective to them, that they're not going to be too burdened, and that it's simple to use and interpret. Now, as someone who had to go to urgent care before AACC or a few weeks prior with an upper respiratory illness, looking to get antibiotics, they had to screen multiple target or multiple indications to determine what it was. Was it COVID? Was it flu? Was it strep throat? All three of those tests were different swabs and different tests, and the bill I got in the mail for all of that was around $600. That could have been done on a single test, and that's truly the value of multiplexing. $600, 3 different swabs to get 3 different results that could have been done in a fraction of the time with a much less invasive method to the end user. Taking manufacturability aside for a second, novel and non-obvious, a great way to establish that is through multiplexing. I mentioned breast cancer and reoccurrence, for example. Now, HER2 is a very common biomarker associated with breast cancer and HER2 specific breast cancer, but that in itself isn't novel, right? It's really well characterized, really well documented. You're not going to get any IP around that intended use. Now, what if you started using multiple markers to establish a new and non-obvious value associated with reoccurrence or predicting reoccurrence? Maybe it provides a score for that reoccurrence by combining, say, HER2 combined with CA15 combined with EGF or VEGF, and all of a sudden you have a value that is novel, it doesn't exist in nature, isn't obvious, and you can start building an IP profile around that. And then the other benefit is manufacturability. So if I have to and want it, all the benefits I alluded to with the multiplexing approach. Now, what's that mean in terms of a cost point to me and well to the end user? So multiplexing is a great way to reduce costs because the assembly, the cassetting, the more labor intensive components will all go down with multiplexing. You also have the opportunity to reduce consumables. If I have to take 3 swabs in the example of strep, flu, and COVID, now I'm just down to 1 swab. All of that are benefits in the manufacturability, so usually costs will go down. The first risk I wanted to discuss is engineering obstacles. So with multiplexing can come the risk of customization. Now, every time I hear the word customization, usually it comes with a price tag and an impact on my timelines or time to market because you're making something new. So what are these kind of custom components of it that might get triggered by multiplexing? The first one is the cassette. So on the top left here, you see kind of a standard blown up view of a cassette. Now, this is a standard 2-line cassette, and these are very common 60 millimeter strips, 4 millimeters wide. The frame of a 2-piece cassette might be 80 millimeters. Now, what if we added 3 or 4 lines or a second strip? All of a sudden, you're kind of deviating from a lot of the off-the-shelf options to kind of really get you to market quickly. So, for example, if I have 3 lines, I'm going to need a wider reading window so I can get all of those on a single interpretable space. I might have to elongate my wick pad to absorb more volume, or elongate my conjugate pad and sample pad for the same reasons. So cassette customization, although pretty straightforward in the solutions to all of these, does take time and cost associated with building something new. And how about the materials that I just alluded to? There's a lot of materials have a custom length that they have, whether it's your sample pad or conjugate pad or membrane. These comes at very standard sizes. Anytime you deviate from that to meet a very specific need associated with multiplexing, because the material itself could be a solution to some of the other obstacles we'll talk about in performance requirements. But the material itself, if you go in the customer route, might take 4 to 6 months for that to be done. Now, cost-wise, your materials are usually negligible, or a few pennies, if that. I think total for most lateral flow strips is 5 cents or less for the paper-based material. But as soon as you step away from that to get custom length, that price is going to go up. What about the usability impact is another one. We talked about a single swab for the upper respiratory example I was giving. What if I needed more volume than that because I'm running a longer strip and 100 microliters wasn't going to work? Do I need to collect more samples? Do I have to sit in someone's mouth longer? How are they going to have to process that sample? There's a lot of considerations with regards to multiplexing to ensure that it maintains that value of usability benefits that we discussed earlier. And the last one is the reader. Can the reader interpret multiple test lines? Not only interpret, but analyze and calculate, apply perhaps a dose response curve to generate a concentration. All of these are considerations because there's a lot of readers that don't have that capability. So what are these performance requirements that I was talking about that materials can help overcome? Well, here's just a list of some of them, and there's a lot more not listed here. Interference and cross-reactivity, that's pretty straightforward, right? Some analytes might be too similar and too analogous to be tested on the same strip. Perhaps IL-4 and IL-6 are too similar, where your epitopes are conserved across antibodies you've selected for your initial target. So you always have to be considerate of what that cross-reactive and interference kind of footprint is for your assay. Assay runtime, as the reading window and membrane get longer because you're developing more tests, the assay is going to take longer to develop. And that's one of the more complex parts of multiplexing is if you're interpreting it at 5 minutes or 10 minutes, all assays have to be completed in that time. Analyte concentrations, we had a complaint or a concern from the NIH for a grant just a few weeks ago about, our 2 targets on our multiplex assay being orders of magnitudes apart, and how are we planning on overcoming that on the single strip. You have to worry about what that, any dilution requirements for one assay is going to impact the second assay. Speaking of dilutions, what about buffer formulations? Each assay is typically, and more specifically the antibodies, have a specific range they want to be in terms of pH, protein levels, salt concentrations. You have to make both assays work within the same formulation, which is not always as simple as it sounds. Conjugate loading, if you have multiple targets on a strip, you're going to have multiple conjugates typically. How are you planning on impregnating the conjugate pad with more and more particles? And then the data result interpretation, is it going to become harder for the user to interpret the results? And I added the example on the right for exactly that. And it's one of my favorite images because there's a whole lot of things going on in terms of why multiplexing can go wrong. For example, there's no control line, right? Not only is there no control line, you can see irregular flow as they're staining on both the left, right of the membrane. You can see, the leading edge effect on the test line with the more intense darker band at the start of test line 1. All these can be attributed to complications with multiplexing. Another risk is manufacturing scrap. Now, with more test lines on a single strip or multiple strips in a single device, you're going to have more and more release criterion and QC requirements. Now, if any one of those strips fail or QC fails on one of those test lines, the whole assay is going to fail. That means you're scrapping functionally both assays, even though one might pass. Switching to a multi-strip approach kind of mitigates some of those costs, but then you lose some of the benefits of manufacturing efficiency because you're still manufacturing 2 strips while still gaining the ability to cartridge only one time and foil one time. So there's benefits there. But you still have this risk of increased scrap. And this is very common because with more test lines, become more opportunity to fail. And the last risk that everyone knows about is elongated timelines. With more complexity is going to be more time. With multiplexing, you're providing more, which means you're to the customer, which also can mean that you're taking on more risk in development. That might surface as elongated timelines, and time is money, so it's a consideration that has to be made early on to ensure that you have enough runway to accomplish this, because the value is clearly there and the benefits far outweigh the risk, in my estimation, for a lot of intended uses. But it has to be considered early on, and make sure you build a plan around that timeline. Speaking of elongated timelines, I think that's a great transition to some of the strategies to overcome these. And the first strategy I have for you is in your biomarker selection. My advice is to go into this with a very open mind and be ready to pivot. And my approach has typically been to evaluate individual biomarkers associated with a particular use case, characterize them particularly clinically, and then tier them. And then once you're there, you can understand the risk and benefits of combining them. For example, if you have your top 1 through 5 biomarkers associated with oral cancer screening, and you find out that your top 2 do cross-react to a degree, you can sit there and play with optimizing the assay or reagents or condition to get it work, or you can pivot to your top antibody and your third antibody. So just be flexible to understand the impact of timelines and going down these rabbit holes of development. Another strategy is on material selection. They always say garbage in, garbage out, and that couldn't be more true when it comes to multiplex lateral flow. Why that's the case is, A, you're going to be scrapping more traditionally with multiplex assays, and the cost of that scrap is going to go up because there's multiple antibodies associated with each of those strips. So make sure you pick the right antibodies, and don't be afraid to customize antibodies. Either hybridomal libraries or recombinant expression, or great vehicles for a lot of different things, including supply chain control, cost mitigation, performance. So don't be afraid of the early short-term cost of that process, which might be 30 to 60,000, for the long-term benefits that I just listed. We are lucky at nanoComposix to have a partner company like Bethel Laboratories who can provide us those services and integrate them into our assay development and product development services here at Nano. What you're seeing on the screen here is their antibody validation pillars and really the development process. They really prioritize characterizing and selecting the right material. Just because you have 100 clones, for example, in recombinant antibody production, selecting the right clones is difficult. And how do you know those are going to result in the performance you want early on, so you're selecting them? That's what Bethel Laboratories focuses on, to really ensure that you have the right products at the end of their development process. Another strategy is going to focus on the timelines. You want to plan accordingly and set expectations. With all the benefits we talked about and alluded to throughout this presentation, it's going to typically equate with a longer time to market, and that has to be understood early on. Ensuring you have that runway to get there, identifying those risks and rate limiting steps, and creating the redundancy in your processes to kind of mitigate the impact of those. We used the custom antibody development as a solution for supply chain control. So maybe you build that in early on in redundancy, so you can have that benefit available when you need it, when you're trying to lock down your BOM and feature design. Taking advantage of the lateral flow platform versatility is another strategy. Don't lose track of your ability to customize the plastics and the housing, the consumables, the strip itself, playing with the conjugate formats, strip geometries. Lateral flow is and always has been a very versatile, low-cost method. Take advantage of that when you're trying to resolve some of the issues that are going to absolutely come up during development. In addition to some of the strategies we just covered to be successful in the multiplex space, I wanted to list also some solutions associated with some common problems. One of which is those products that need semi-quantitation or fully quantitation in their outputs. Having a reader and the right reader is going to be important, and there are readers out there capable of detecting multiple test lines on multiple strips. For example, the Cerebus reader from Lumos is one example, and these are the images you're seeing here are the trace outputs and the device footprint itself. A common obstacle in any assay, multiplex or not, is going to be sensitivity. And typically when you are multiplexing, you're going to have to sacrifice things like dilution and formulations. So overcoming some of the impact of those on sensitivity can be overcome with the right nanoparticles. So our backbone at nanoComposix is in those nanoparticle solutions. We develop a gold nanoshell for precisely this reason. Gold nanoshells are quite a big step away from the 40 nanometer gold that's ubiquitous in this industry. These particles are designed with their optical properties in mind to be much more brilliant on a test strip than 40 nanometer gold. These are silica cores shelled in gold, so it has the fluid characteristics of small particles, but the optical properties of large particles. We found that in the right situation, they can provide quite the benefit, up to 20-fold increase in sensitivity. Now, that's not every assay, and it's not a silver bullet solution to a problem, but I highly recommend when you're trying to overcome specific obstacles associated with sensitivity. A great example of overcoming some of those sensitivity issues with the gold nanoshells is with the troponin I assay. High sensitivity troponin I, because of its intended use and the risk associated with misdiagnosis, high sensitivity is required. Here's an example of 40 nanometer gold versus 150 nanometer gold. In this assay with these antibodies, you can really see the difference in signal intensity in the plots provided. So bringing all this back home to end the presentation, I wanted to bring up the Vigilant Biosciences example again. This product that we developed with the team at Vigilant had all of those complications and risks that we identified and talked about in this presentation. We did have technical considerations that we had to overcome with switching to a multi-strip format. We did have engineering obstacles and customization was required. Knowing that up front, that we're able to properly plan and execute our development strategy to successfully get a product to a point where the FDA is very excited about this and gave us that breakthrough designation and working with us through that submission now for an OTC or POC test capable of doing oral cancer screening in saliva that is fully multiplex and fully quantitative. With that, I'm going to wrap up today's presentation. I want to thank you all for joining me. Again, I'm Andre Alfaro, and feel free to reach out to us with any questions you might have about this presentation, multiplexing, or assay development and reagents in general. I definitely recommend reaching out, and I'd love to talk to you more about what you guys are working on and possible solutions that we can provide. So thank you again.
Thank you, Andre, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you'd like to ask, please do so now. Just click on that Ask a Question box, type in your question, and click Submit. We will answer as many of your questions as we have time for. So let's get started. Andre, we've got some great questions already coming in. I'd like to start with this one. What are the considerations for improving sensitivity in a multiplex assay?
Great question. I would break this up into three considerations. Reagent-specific considerations, assay-specific considerations, and platform considerations, each with very different avenues for improving sensitivity and performance. On the reagent side, in a multiplex assay, there's nothing more important than the affinity reagents themselves. How they interplay between each other and how they bind the target analyte is going to give rise to functionally most of your analytical and clinical specifications. So, reviewing and evaluating alternative antibodies or commercial antibodies and proteins is a good place to start. You can even go down the route of custom antibody or antigen production, really tailored to your specific need. That is on the reagent solution end if I wanted to improve sensitivity. Now, if I go down more on the assay specific limitation, it's a bit harder, mainly because those are predominantly associated with your target matrix and the clinically relevant range of that protein in it. That's not going to change too much, maybe a little bit on your volume per test or any dilution requirements that you have. You're really looking for more of a reagent solution or tool-based solution to improve sensitivity. Those are things like gold nanoshells that improve sensitivity based on just plasmonics. So something that can enhance sensitivity without changing relevant concentration ranges. Those are kind of the tools you're looking for there. And then there's platform changes that you can make. Lateral flow has a lot of value, but it does come with some limitations, particularly around total volume. You could innocuous to test with, assay runtimes and kinetics. People are not going to buy a 60-minute lateral flow test. They want that thing to be, 5, 10, 20 minutes at the longest. So, if I'm trying to improve sensitivity, given that some of my limitations for sensitivity might be volume, might be assay runtime, maybe I engineer a device specifically to overcome those obstacles, or changing the material in there that have different pore structures, different bed volumes to take on more samples, so thus more analyte and improving sensitivity. Maybe I slow down my assay to improve my binding efficiency at the test line or the conjugate. Those are different engineering changes and material changes you can make to really improve sensitivity. So, excellent question.
Great, thank you. Our next question: how is cross-reactivity addressed in the optimization process?
Oh, yes. A question we get a lot, mainly because there's always going to be levels of cross-reactivity and interference. That is just the nature of the beast typically. And really it starts with really understanding the root cause of that cross-reactivity. So typically that begins from an R&D perspective on deconstructing that device, really understanding the subcomponents of your assay and how they contribute individually to the observed result. Typically, taking out the conjugate pad and putting that into, a different environment away from your membrane or your sample pad, really isolating the component and testing it to see where the issues result from. Is it aggregation in the conjugate pad? Is it cross-reactivity of the antibodies at the test line? Is it membrane or material specific effects? Until you deconstruct it, you really don't know what's contributing to it. So that's where it starts from. And I think you should all be prepared for the unfavorable answer to this, which is it's usually a combination of multiple variables that contribute to something like cross-reactivity. And there's typically not a silver bullet solution unless you identify really it is an antibody whose epitope is conserved across multiple of your analytes and causing some non-specific binding or potentially some interference. But we'll ultimately the response to this is first isolate and identify what's contributing on that pie chart, what percentage to the problem, and coming up and devising really specific solution to those problems. And that's one of those things that just comes with experience. And after this presentation, if you have any other questions on specific examples you're interested in, just shoot me an email. I'm happy to go through this in more detail with your specific issue.
Thank you, Andre. Now, are there any specific considerations for different sample matrices?
Oh, good question. I think matrix-specific problems, there's the fundamental pathway to develop multiplex assays kind of independent of matrix. There are, however, matrix-specific issues you have to overcome, but that's kind of independent whether it's multiplex or a singleplex assay. It always boils down to the cornerstone of a multiplex assay of can you detect multiple analytes and function in the same environment as other analytes? Same dilution, same buffer formulations. This way you can conserve costs, provide more information. There's a ton of value to a multiplex assay. But the matrix-specific considerations are typically things like, how are you going to collect that sample? How are you going to move that sample? And that's, independent on whether you're using a multiplexed assay. That being said, some matrices are inherently harder to develop assays in, period. Something like saliva with its difference in viscosity and pH and all sorts of considerations for how you're going to collect it, what part of the mouth, what time of day. These are all variables that impact not only the sample matrix itself, but also the relevant concentrations of those proteins. Other things like blood, which is highly conserved, fixed pH. Specific gravity is going to be the same. There's not too many matrix issues to overcome. However, you're dealing with volume limitations There's a lot more proteins that might cause potential interference. There's hemolysis and leaching that can interfere with the assay. So there's quite a bit of differences in the different matrices, but overall, with regards to multiplexing, I think your cornerstones stay the same for what you're trying to accomplish, and that is, the clinically relevant ranges for my assays detectable at puncture the same dilution and the same buffer and at the same sample volume, and those things are very much specific to any assay.
Great, thank you. Next question, and we have time for a couple more. Are there any differences in quality and regulatory requirements between single analyte and multiplex test?
There is, and quite a bit so. I probably need permission from our legal team to answer this in any great depth, because we're not a regulatory firm. But I can speak to this through experience with the FDA, with regulatory bodies like Health Canada and IVDR. When you have a multiplex assay, you have multiple products functionally. And yes, the submission might be a single product you're submitting for the LFA test strip itself, but each of those assays will have its own specifications. So each of those assays will have its own LOD and precision and linearity, stability, time to result, all the analytical testing thing on top of your clinical ones. So it's quite a bit of work. Functionally characterizing each of these assays, standardizing each of these assays, and how they work individually as well as with each other. I've gone to the FDA with multiplex assays that were fully quantitative, and those values were combined. How are we combining them? What is that score? What is that algorithm? All of that was part of the submission. So quite a bit of regulatory considerations to think about when you're doing this. It does inherently make it, in terms of time, if you're doing 3 assays you want to multiplex together and submit as a single product, 3 submissions versus 1, less hours of work but more risk. So always consider that, because if one fails, it all fails. This is very true particularly for assays where you're combining scores, where if I fail QC or functional results on one test line, all test lines fail, regardless of their output, whether they're in spec or not. And the FDA knows this, and these regulatory bodies know this. So they pay close attention to these things, during submissions and during reviews. So you definitely want to set up your pre-subs and your Q-subs to understand what they're expecting, and then really tailor your development with regulatory and commercial in mind. I think that people can sometimes lose track of where they're going as they're iterating through development, but really understand what the product specifications you want is, what that intended use is, and how you're going to use this device to meet that intended use. So I'll leave it at that, but that is a much more time would need to be allotted to answer that one in greater depth.
Certainly. And to our audience, we will be accepting on-demand questions. So if there's any questions that we don't get to today or during that on-demand period, just keep submitting them. They will be answered via the contact information you provided at the time of registration. So Andre, I think we can squeeze in 2 more questions. Let's go with this one. And this one has come in a few times in a few different variations, but when would you select automated versus manufacturing solutions?
It's a good question and a simple one at its heart, but it has a kind of a long-winded answer that I'll try to get to as quickly as I can. There is no exact cutoff with manual versus automated solutions. There's benefits to both. We use automation down to lot sizes of 5,000 units per lot per run. But we also use manual manufacturing for up to 10,000 units. So you might ask, okay, why manual over automated at those volumes that you can do both? Because there's a little bit of a give and take with that. So typically for development, as you're iterating through your assay until you really hit best mode freeze, not a design freeze, but you've functionally made a prototype that meets all your specifications. That iterative process is typically at a manual or card-based manufacturing approach. So inherently, the time to transfer that technology to manufacturing, develop a processing QC around it, is inherently faster since you've been doing it manual from the start. You really don't integrate automated solutions until you're kind of in the meat of product development as you're closing in on a target product profile. So that being said, if you have the time to invest in moving from card-based or manual to automated, you get a whole new ladder of pricing. Card-based or manual-based manufacturing does not scale, right? There's a time it takes to make one card, and that card is just, there's some cost savings, assuredly, but functionally, if it takes, an hour to make 10 cards, it'll take 10 hours to make 100 cards, and those costs are usually 1-to-1. Automated, you have very much a steeper slope. So the cost differences between, even 10,000 and 20,000 might be, 40% cost reduction. The difference of manual manufacturing 10,000 and 20,000 units is just, it'll be twice as expensive, or it'll be the same price per strip. So, if you have the time, then I would always suggest going on to an automated solution, even at volumes as low as 5,000. And why? It's because it opens the door for more volumes in terms of scale. So this way, when your commercial need exceeds that and you want, 10, 20, 50,000 strips and you want that cost associated with higher volumes, you're really not going to get that with manual-based manufacturing. You really only get that automated. So if you can, that old adage of buy once, cry once, if you have the budget and the time to do it, you now have a pathway for meeting cost points on manufacturing, which you really wouldn't have. But again, path is a little slower because you haven't been starting with that kind of process and equipment from day one like you were for manual-based manufacturing solutions. So I'll leave it there. But yes, it's very much product and project specific, and there's benefits again to both sides.
Thank you. That was a great explanation and really great examples for us to easily understand that. We have time for one more question, and let's go ahead and use this as our last question. Are there limitations to how much multiplexing you can do?
There are limitations. I live in a world where nothing's impossible, but sadly is just not true. They're just usually impossible. Just comes with time and money. It is the two most important variables typically when you make a product anyways. So I've been asked this quite a few times, can I go do 5 assays, 6 assays? Popular is the respiratory panel with COVID, flu A, flu B, RSV, plus you have a internal flow control line, maybe you have a negative control line. Can you have 6 lines? Can you have 8 lines? It always comes down to the cost to do something like that. It is not impossible by any means, but you have to have a lot of things work in your favor. And it inherently has limitations. You think about the volume and the flow across this assay, the amount of conjugate that they put into the conjugate pad. You need all these lines to develop and stabilize at a fixed time. A typical reading window on any lateral flow cassette is 10, 20 millimeters. And each of these test lines are spaced at 4 or 5 millimeters apart. You're going to have a 40, 60 millimeter reading window. That sample has to travel that whole distance and migrate through that path. The kinetics and binding kinetics have to develop those test lines and stabilize, because that flow is going to get slower as it moves farther away from the conjugate pad. How do you overcome the kinetics issues? How do you get enough volume in that assay where it doesn't flood the strip and go over the material instead of through it? And how do you impregnate the conjugate pad with conjugate specific to all these test lines? Because otherwise you're going to get a whole lot of aggregation. So there are some technical obstacles to overcome, and if you can overcome them, there's really no limit to amount you can do. So it's just really specific to what you're trying to accomplish and how much budget and time you have. Anytime you hear custom, anything. And that's typically when you do more than, say, 4 lines. If it goes down this custom route, now you need a bigger custom cassette, custom materials. That just takes time. Takes time to develop those, takes time to source that material. And with that in mind, you can make that decision yourself whether you want to go down that route. Now, there is a inflection point where the cost and risk kind of invert between too many lines, because then again, going back to the regulatory consideration, if one assay fails, they all fail. So if you have 6 assays, all it takes is one of those to fail and the whole strip fails. So imagine the cost to validate something like that when you're throwing away countless amounts of expensive reagents because one assay has not specific binding because the new lot of your X antibody was sticky. So there's a lot of gray area to this question, but I would say no limitations, but expect longer development times and more cost as you move away from more standard designs with, say, 2 target analytes. As you move farther from that, expect that time investment and cost investment to be higher.
Andre, thank you for this excellent final comments for our audience.
Good question. Not from my end. If anything, I feel like I didn't give these questions any level of detail I would like to. So feel free to reach out to me directly after this meeting. I believe my email somewhere associated with the presentation, but not in the presentation itself. So feel free to reach out. My job is to really walk through these questions with our customers and help them understand what product development is, how to use lateral flow to overcome or meet a product need. So don't hesitate to let me know if you have any other questions, or even to get into these questions in any more detail.
A big thank you again, Andre, for your time today and for your important research. I'd also like to thank LabRoots and our sponsor, Fortis Life Sciences, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today and for their interesting questions. Like Andre mentioned, questions we didn't have time for today, and additionally, the ones you submit during on demand, they will go straight to Andre. So please send them in, and they will be addressed by Andre via the contact information you provided at the time of registration. Now, today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Thank you so much again, Andre, for joining us, and to all of our friends out there. Until next time, have a great day.